My name is Carroll. I'm a distinguished professor of biochemistry. I would describe myself as a molecular biologist or molecular geneticist. I do work on genome editing. That's what the field is now called. I've been at the University of Utah for 41 years, and in that time I've been doing research, teaching, administrative work. I get a great deal of satisfaction out of even the smallest lab experiment that works and tells me something that is an answer to a question that I've asked. Genome editing is a description of technologies we now have to make very specific changes in the chromosomal DNA of essentially any organism. And the secret to this is that we have ways to make very specific, very targeted breaks in DNA. And this sounds like a bad idea. Break DNA doesn't sound good. But it turns out that cells have ways to repair those breaks in DNA, and we take advantage of those cellular processes to make the types of changes that we're interested in. We have several types of what we call programmable nucleases. And I started working now almost exactly 20 years ago on the first of these platforms, and these nucleases are called zinc finger nucleases. Genome editing technology is being used very broadly. People are using it for just investigating the natural world. You can modify a gene in a fruit fly that corresponds to a gene that we're curious about in humans and in other organisms, and in the fruit fly it's easier to discover what the function of that gene is. Make a mutation in it, discover what the consequences are. Distinguished Innovation and Impact Award, I think, recognizes the fact that just learning how the world works can develop into tools that we can use. In science, one tends to labor in obscurity for kind of a long time, and to have people acknowledge that something you've done has had an impact is gratifying.